So we are continuing what we just were talking about, regulation of the cell cycle. I'm going back a little bit because I needed to kind of restart. Here we go. So we know that this is the cell cycle. You can see it here on the right. And we talked a little bit about this idea that as a cell is moving through this in its life, during its lifespan, where it's growing and doing its normal job, and then some signal will allow it to go into the S phase of interphase right here, where you see DNA synthesis. But this, before it can go in there, it needs to pass this thing called a restriction checkpoint. You see there in the little square there below. And so there will be a signal that will allow that to happen. And we want to talk about what that means coming up. So, but what we do know is that in normal cells, this cell will not move into making copies of its DNA until it's been allowed to pass through this checkpoint. Once it does, it goes into this S phase of interphase, makes the copies of its DNA, and then if it passes, again, there's actually another checkpoint here that's not shown, it will be allowed to move on into G2. And once it gets to G2, here we have another checkpoint. And then even in mitosis, right, during the metaphase of mitosis, there's another checkpoint. So all of these things are to control, regulate the cell cycle so that when we get the new daughter cells that would be formed from that, they're in the, everything is the way it should be. So we don't have any mistakes made, any errors that would cause a problem with the next generation of cells. So that's more or less what these checkpoints are about. Now, to regulate the checkpoints is what we're going to get into. And what we're going to look at is these two kind of factors. One is that there can be things from the outside of a cell, these external triggers, which can be sent in via some type of chemical messenger into the cell to either initiate or start it moving forward in the cell cycle, or to inhibit it. So these would be some of the things that could be a part of that external regulation. So if the cells nearby have died, if there's a release of certain growth hormones by the organism, or if there's too much crowding of cells, all of these things could be considered external triggers that would then send messages into the cell. There are also some internal factors, and we're going to look at that too. So let's go forward. So at G1, we know that the external factors are going to be stuff like, do we have everything that the cell needs? So adequate cell reserves. How about the size of the cell? If we lose some cells around this cell that we're talking about, that might mean that the cell is going to need to replace itself, or we need to fill in a space. So those things are external influences. Some other things that we would have to be sure of at that G1 checkpoint is that the DNA is in good shape. If those things are met, if there's enough cell reserves, in other words, enough materials in the cell cell size is of normal size, and we have no DNA damage that's detected, then the cell could go into the S phase. If it doesn't, then it won't go in the S phase, and we're going to look at how that occurs. We mentioned that if that happens, we would stop the cycle in some way, and we'd enter something called G0. So remember, G1 is where we're at right now. This is like a holding pattern until we figure out whether or not the cell can make a copy. G2, we're going to look at the DNA that's been replicated there. Is it all in good shape? Is it the chromosomes are all exactly as they should be? So we have exact copies of the DNA. The cell can inhibit the G2 from going forward if there's some error. But there's also some safeguards in the S phase that won't allow it to move on in that synthesis phase if there's a problem. So there's a couple of different things that the G2 checkpoint's involved with. One is being sure that everything is in good shape, that we have all the materials we need, and then did the DNA that came from the S phase, is it also as we need it to be perfect? Any problems, you're going to stop that cell cycle at that point, see if you can fix it. M phase, checkpoint, metaphase, remember we looked at this a bit, where we said that metaphase is where all the sister chromatids, in other words, the chromosomes plus their copy, which are attached at that point called a centromere, and then have these little hook-like structures called kinetochores on them that are attached to spindle fibers. So that all has to be lined up and all correct. If it's not, then the M checkpoint during the metaphase of mitosis will not continue. At least in theory, it should not continue. So we want to be sure that that wouldn't happen so that you didn't end up with some of those sister chromatids ending up in the wrong place. So let's look at the regulators. That's where we kind of left off. Positive regulators are going to move a cell into the next step of the cell cycle, and negative regulators are going to stop the advancement of the cell cycle until we figure out what we need to do. Now these regulators, let's see if I can expand this a little bit, but what those regulators are involve proteins, of course, things called cyclins, and some cyclins that have some specific enzymes called kinases, called cyclin-dependent kinases. Those are the enzymes that are working with these cyclins. So when these different cyclins, which we went through last time in the earlier video, they will be made by the cell. This is genetically programmed so that there are certain things which are going to trigger the production of cyclins at various stages of the cell cycle. And then that is also going to influence what happens from there. So thinking about those things we talked about, internal triggers and external triggers are going to be involved with promoting this production of cyclins. And then the cyclins will then have an effect, and we'll see that. So if it's a positive regulator, then what that's going to cause is, let me see if I can make this a little bit better there so you can see it all. So a positive regulator would have the production of a cyclin, and what we would see there is the cyclin would bind with these things called cyclin-dependent kinases. So here's the cyclin, here's a CDK, or cyclin-dependent kinase. They bind, and once they've bound, then what we get is a phosphorylation. So we get a phosphorylation. Remember, it's a kinase, so we're going to have something to do with a phosphate. We're going to get phosphorylation of this complex. Once it's phosphorylated, it's going to go to some target protein. I think this is kind of where we left off last time. And then that target protein, it will transfer that phosphate onto the target protein, which then is going to activate that protein. So let's see what kind of proteins we're talking about. Here's some, so there are a number of regulators, and these are the negative regulators. So remember that a negative regulatory molecule is something that's going to inhibit the cell cycle at that stage until we're sure that things are the way they need to be. So here's one of them. So there's actually several of them. There's something called RB, and RB stands for this molecule called retinoblastoma molecule. It is something that was recognized via that condition, actually a disease condition called retinoblastoma. But what is understood is this retinoblastoma protein is a regulator. It's a negative regulator. So it's going to stop during the G1 checkpoint. It's going to have an effect on the cell cycle going forward. So and we're going to see that right below. There's also two others, P53 and P21. So let's start with this RB regulator. So RB is here, And when RB it's in the cytoplasm of the cell, it's during, and it's actually involved with the DNA itself. So this RB is at this moment not phosphorylated. And when it's not phosphorylated, it can bind to this thing called a transcription factor. And this particular one we're saying is E2F. And if RB is bound here to E2F because it doesn't have a phosphate group on it, then it won't allow for any of this part of this DNA to be transcribed. In other words, to be copied, just that one part. Okay, and that's important. But if it gets phosphorylated, so remember our CDK cyclin event we just mentioned, if it gets phosphorylated, then what we see is that in that case, that phosphorylation of this complex releases RB from this E2F. And that's going to then bind to the DNA, and it's going to turn on some gene here on the DNA that's going to then create some type of protein. Okay, so that's RB regulating it. So RB is regulating this process. When it's not phosphorylated, it's inhibiting the process from going forward. When it gets phosphorylated, then that inhibition is released, and therefore then that can cause some activity. So some gene is going to get expressed. Some other protein is going to be made. Now I'm going to show you this using another website here in a minute because it's really helpful to see that. And so I'm going to shift, if I can, let's see if I can do this, to this right here. We're going to do a little of this in class. So this is another way to look at it. And I like the way that this website, this is the Howard Hughes Medical Institute who give us this particular image and this activity. So here's cell cycle, you recognize it. Here's the interphase, G1, S, G2. Notice that they did put a checkpoint into the S phase. So we're looking at, first of all, G1 during interphase. And we can look at this cell cycle and understand that this is a checkpoint that is going to determine whether or not this cell is healthy enough to move on into the S phase. So here's a little short checklist summary. If there's no DNA damage and there's enough resources, then this cell can go on into the next phase. So that's that G1 checkpoint. So we're going to go back here. We're going to go through this S phase checkpoint. We just look for no errors in the DNA, so we're good. We go to G2 checkpoint. Okay, again, another double check. DNA no damage, chromosomes are all there. We have enough of the other materials we need to proceed, such as enough ATP, enough of the organelles that are going to be needed for when we divide this cell into two. So there we go. And then here's the mitosis, or the M phase checkpoint right at that metaphase. Is everybody lined up correctly? Is everybody attached properly to the spindle? So that's the phases, and those are the checkpoints. Now, we're going into the regulation part. And so if we do this, what we see is that we're going to have regulators that are either going to cause the stimulation of the cycle, in other words, they're going to promote the cycle going forward, or they're going to inhibit the cycle from going forward. Now, the proteins, these stimulating proteins, are also called something they're encoded by these genes called proto-oncogenes. Now, oncogenes is kind of a confusing term because onco has to do with cancer. So when we look at proto-oncogenes, we're really not talking at this moment about cancer. We're talking about genes that are allowing certain things to happen. And all of these are going to be important and are regulated by those things we mentioned earlier, those positive and negative regulators the things which are CDKs and cyclins, CDK cyclins. So we're going to see how this works in this cycle here. But here, when we're looking at this, we're talking specifically at the genes within the DNA itself that are encoding for these different regulating proteins. So we're going to see how that works. So let's start here with G1. And if we click on this arrow right here, what we see is that in G1, remember we have CDK cyclins are going to move, the positive regulators that are going to stimulate the cycle are going to move this cell into the S phase. So these positive regulators could be growth factors, things that are telling the cell it's time to grow, it's time to produce more cells. So in this case, we would have the CDK, the cyclins bind to CDK, and then do a phosphorylation to drive this cell into the next phase, which is the S. But then there's also these inhibitors. And so now we're looking at the inhibiting proteins. So in this one, what you're seeing is that we have a protein called P53. And P53's big job is to check to see that the DNA is perfect, that it's not damaged. And if P53 sees any damage, it's going to stop any activity. Even if there's a growth factor that comes in, it's going to stop the activity. It's going to inhibit this CDK cyclin complex. It's not going to allow that phosphorylation. It's going to stall this cycle so that it cannot move forward until it can be determined, can the DNA damage be fixed? And so it's going to put this into G0. So you notice this over here, G0. It's going to put this cell into G0 until it can be determined if the DNA can be fixed. And if it can't be fixed, then the cell is going to undergo a process called there's the word apoptosis right here, which is going to be the cell will then self-destruct. So we're going to get rid of that cell because it has DNA damage that cannot be repaired.